Hey y'all, this is Tolbert. Just putting this together especially for y'all so that you could watch this and do what you need to do in order to help yourselves as we embark into you guys being more able to handle your futures. I'm putting this out to you so that you can do this piece on your own as well. This will be the last bit of notes that we have for Chapter 8. We're gonna talk about what's the Calvin cycle and finish it all up for you. So what's the Calvin cycle? Calvin cycle is part of the dark thing, right? In class, we talked about the light part of this cycle in photosynthesis, and the light part is, of course, where the oxygen is being made. So you've got water being split in the oxygen we can breathe being made. Now, the dark part happens, and the light part happens in these happy green pancakes, these thylakoids. And the scissor up is the stroma. So the stroma is the dark cycle where the Calvin cycle happens. So carbon atoms are bonded or fixed into organic compounds. So as I was saying in class, this is where it snatches these carbon, because remember, in order to make sugar, the first part of the molecule, C6H12O6, the C6, you need 6 of them, all right? So the Calvin cycle has 3 main steps occurring inside the scissor or the stroma. This example you should probably write someplace, like if your finding sheet is around or someplace that you can study it. In your book, and also I mentioned this NADP is a carrier molecule. NADP is exactly that. It carries these high energy electrons and moves them around. Your book gives the example of this oven mitt, and you reach into the oven with your oven mitt. It's going to help you transfer that hot potato from the oven to the plate. That's what NADH does, NADP plus does. We add the H to it, we add the little electron, and it's going to grab onto it and transfer it to where it needs to go. So what exactly is ATP? ATP is the energy currency for the body, for cells. It's the actual energy that the cell's going to use to do all the fun things, which is called living. We like to live. It's made of adenine, ribose, and 3 phosphates. Thus, it's a triphosphate. Okay. In class, I would show you this, but I'm going to tell you to go ahead and find that video on your own. So what are the steps of photosynthesis? And when is ATP actually made? That's a great question. Let's get into it. First step, the carbon dioxide diffuses into the stroma. Remember, the stroma is the scissor around the happy green pancakes from the striding cytosol. The enzyme combines with the CO2 molecule and a 5-carbon carbohydrate called RUBP. Then it keeps on going and it makes something called PGA. The second step is the PGA molecule receives one of them phosphate groups, or the electron that oven mitt has moved, that hot potato, and it's going to move it and do some work with it. That's the second step. The third step is the molecule PGAL is converted back into RUBP. And the PGAL leaves the Calvin cycle and is used to make other organic compounds like amino acids, like lipids, carbohydrates, all kinds of things. PGAL serves as the starting material for the synthesis for glucose, fructose, and all these toasts. Remember, glucose, that end of that word means sugar. Glucose, fructose, maltose, sucrose, all of those are sugars. Here's a great picture, just kind of following along where things are going to happen to the stuff that's going through in photosynthesis. You've got the roots here sucking in that water and other vital nutrients, and that water is going to be transferred up there. And in the first stage of the light reaction, it's going to split that water, and oxygen is going to be liberated. And then the second step, of course, the dark reaction, the Calvin cycle is going to all happen. It's going to be nice there. So in the leaves, that's where you're going to first find that sugar. Okay, that's C6H12O6. So glucose is also a monomer used to synthesize polysaccharide starch and cellulose. Each turn of the Calvin cycle is going to fix or hold on to one CO2 molecule. It's got to take 6 turns. Why 6 turns? Because it is C6H12O6. So 6, you need 6 carbon molecules to get it done. Photosystems and electron transport chain. Only 1 in 250 chlorophyll molecules convert this into usable energy. A lot of it is used for other mechanisms that we all are unaware of, but it's pretty cool if you ever want to look into it on your own. Other molecules like chlorophyll B, C, and D, or these carotenoids, they're absorbing all these other wavelengths of light that are going to be used for things during the process. And there's a number of other pigments that are known to be there and are doing specific parts during this reaction. So some of these extra pigments are in photosystem 1, some are in photosystem 2, absorbing the light and doing the things that it needs to do. Now, photosystem 1 and photosystem 2. Let's talk about that. Photosystem 1 actually happens second. Now, because we didn't know really about what was happening in photosystem 2, so we named it photosystem 1 before we knew something happens before in photosystem 2. Okay, and your findings sheet, go ahead and make sure you note this particular information because it's going to help you. So photosystem 2 is actually where this carbon fixation happening, or the water's being split and all that. Then it goes down the chain in order to do photosystem 1. Energy is absorbed by this P680. Guess what? 680. It's a wavelength. Remember, these pigments, these buckets that we're dropping this, they contain certain amounts of light energy that they're going to pick up. There's one that's called 680. There's one that's called 700. So each of these have specific frequencies of light that they're going to really cater to. The 680 in photosystem 2 loses this electron and is positively charged, so it now uses that energy to break the water molecule, H2O, split off that oxygen, and thus becoming 4 hydrogen and 2 extra electrons that we're going to use to do all the fun. So these excited electrons, sometimes they have to be replaced or re-excited by the sun. Guess what? And we're going to be doing that and moving them around. Remember that oven mitt, the NADP? That's the oven mitt. It's going to be grabbing these electrons and moving them, and moving it here, moving it there, and releasing them. That's because there's work to be done, and it does be from NADP gets what we call reduced to NADP+. Remember, when you add electrons, it's called reduction because electrons are negative. Let's look at that one more time. So this one here, NADP+, it doesn't have the additional electron. NADPH has the additional electron. When we add it to this, add the H, we're adding electrons. Electrons are negative. That is a reduction reaction. And that is it for today. Hope you had a great time with this, and that's all I need to say for today. Let's go ahead and end it. Where's the ending? All right.